Hello, everyone, and welcome. My name is Rula Deeb. I'm a senior principal at Geosyntac in Oakland, California, and the coordinator of this webinar series on behalf of SERDP and ESTCP. My colleague, Dr. Jennifer Nyman, and I have the pleasure of facilitating today's event. This webinar focuses on DoD-funded research to better understand and quantify the abiotic degradation of chlorinated solvents. First, Dr. David Friedman from Clemson University will discuss the development of a laboratory protocol using a carbon-14 assay to quantify the degradation rate constants of chlorinated solvents based on the accumulation of 14C labeled daughter products. David's presentation will be followed by a brief Q&A session. Then Dr. Dimin Fan from Geosyntac will talk about the development of a field kit capable of measuring the oxidation reduction potential of aquifer sediments in situ. Dimin's talk will also be followed by a Q&A session, and we will conclude the webinar with a longer Q&A featuring both of today's speakers. The next several slides provide instructions on optimizing your webinar experience. If you have not done so already, please download Zoom at the link shown here and provide it to you in your webinar registration confirmation email. If you're unable to download Zoom, you can view the slides using a compatible internet browser, such as Firefox, IE, or Edge, and by creating a free Zoom account. If you continue to have difficulty in viewing the slides, or if your screen freezes, try keying in Control and F5 to perform a hard refresh. If you're accessing the audio through your computer, click the arrow next to the Join Audio button, select Test Speaker and Microphone, and follow the prompts as they appear on your screen. If you continue to experience difficulties, just call into the conference line shown here and provide it to you in your registration confirmation email. You can submit a comment using the chat box related to any additional technical difficulties, but please use the chat box only for comments related to technical difficulties and reserve the Q&A option for questions for the speakers. Keep in mind that you can download a PDF of the slides from the webinar webpage and call into the conference line to follow with the audio. Also note that we will be live streaming the webinar on the SERDP and ESTCP YouTube channel at the link shown here. Just a few comments about how to ask questions. Today's broadcast will be listen only. You can submit questions by using the Q&A box on your screen. You do not need to wait until the Q&A period to submit your questions. Please submit them well in advance of the Q&A sessions. And when you get them in, please add your organization name at the end of your question if you would like us to identify it. The next few slides will provide a quick overview of SERDP and ESTCP. For those of you that don't know the acronym, SERDP is the Strategic Environmental Research and Development Program. It was established in 1991 by Congress as a partnership between the DoD, the Department of Energy, and EPA. SERDP's mission is to identify and address high-priority environmental science and technology opportunities that focus on DoD requirements. And SERDP funds both fundamental research as well as advanced technology development that ultimately impact real-world environmental management. ESTCP is the Environmental Security Technology Certification Program, in where innovative environmental and energy technologies are demonstrated. ESTCP investments capitalize on past investments under SERDP or other research programs and are designed to transition technologies out of the lab and into the field. Especially important in all ESTCP demonstrations is the ultimate transition to implementation and, more importantly, regulatory acceptance. SERDP and ESTCP are complementary programs with much of the SERDP research occurring at the lab and pilot scale, with occasional field efforts, while ESTCP demonstrations are primarily at the pilot and field scale, although occasionally they do support lab efforts. There are several environmental drivers for the type of work funded by SERDP and ESTCP, with the underlying objective of sustaining DoD ranges, facilities, and operations. This, as you can imagine, is a broad undertaking. It takes the form of looking at maritime sustainability, threatened and endangered species, climate impacts, unexploded ordnance, and munitions constituents, as well as other environmental drivers. One key environmental driver is the reduction of current and future environmental liabilities. This involves addressing contamination from past practices, including impacts to groundwater, soils, and sediments. And that's the topic of today's webinar. It also includes addressing UXO contamination and developing management approaches for contaminants of emerging concern. The second part of this is pollution prevention, with a focus on eliminating likely environmental pollutants or hazardous materials in manufacturing, maintenance, and operations on DoD installations. We have several main areas for research and demonstration at SERDP and ESTCP, and these are shown here. And again, today's topic falls under installation restoration. And then technology transfer, it's a very important initiative within the SERDP and ESTCP programs. Technology transfer efforts include the development of videos, training workshops, and guidance documents, with the webinar series serving as a substantial component of SERDP and ESTCP technology transfer efforts. This webinar series highlights research and demonstration efforts from all of the program areas. Upcoming topics include advances in waste-to-energy demonstrations, fuel characterization and modeling for understanding fire behavior on military lands, parameterizing munitions mobility and burial in riverine environments, and as you can see, a lot of other topics. Note that registration is open for webinars throughout 2022. You can find additional information about upcoming webinars at the link shown here. Also keep in mind that past webinars are archived and can be accessed using this link. So you can download PDFs of the presentations from past webinars, and you can also, you're able to view YouTube videos of past webinars dating back to October 2014. To conclude, I would like to remind you that a copy of the presentation of today's session can be downloaded from our webinar webpage. We hope you enjoyed today's webinar, and at this point, I'd like to turn this over to Dr. Nyman, who's going to introduce our speakers. Jennifer?
Thank you so much. It's now my pleasure to introduce our first speaker, Dr. David Friedman, who is a professor and chair in the Department of Environmental Engineering and Earth Sciences at Clemson University. David started his academic career at the University of Illinois before moving to Clemson in 1996. The main focus of his research is the biotic and abiotic degradation of groundwater contaminants, including many halogenated organics and more recently 1,4-dioxane. David received a bachelor's degree in science and environmental change from the University of Wisconsin, Green Bay, and master's and doctoral degrees in environmental engineering from the University of Cincinnati and Cornell, respectively. David, please proceed. And David,
there we go. I had to find the unmute button. Thank you, Jennifer, for the introduction, and thank you for the opportunity to share some of the results that we're getting from SERDP-sponsored research. Good afternoon and good morning to you wherever you may be. So what I'm going to talk about today, I'll give you some background, some objectives, the technical approach we're using, some results, conclusions, benefits to DoD, and next steps that we're working on. So by way of background, abiotic processes as they relate to chlorinated Ethenes has been around since the 1980s, along with biotic processes. I think it's fair to say that interest in biotic processes has dominated up until the recent past, in large part because the biotic processes have been faster and easier to document under the right conditions. But interest in abiotic processes has resurfaced over the last couple of years. And I think we're going to find more and more that they will be useful in reaching attenuation of large dilute plumes, which I think bedevil a number of you out there in your search to turn off the pump and treat systems. In order for us to move forward, however, there are some significant knowledge gaps that remain that have to be addressed. Things like how do we demonstrate whether or not abiotic processes are occurring? That's especially challenging because the products that are formed from abiotic processes are often difficult to discern from background levels. And also, how does mineralogy of the subsurface relate to the occurrence and rate of abiotic degradation? I think before we can begin to realize the full potential of abiotic processes, we're going to need to better understand how it works. So that brings me to the objective of the SERDP project that we're working on currently, where the overriding objective is to understand how magnetic materials mediate abiotic degradation of chlorinated Ethenes in aquifers. The sub-objectives are to develop a carbon isotope assay, aka a C14 assay, to enable us to measure abiotic degradation rates. And then we want to apply that assay to measuring abiotic degradation processes in real water supply aquifers. We also want to assess the proportion of abiotic degradation that's attributable to magnetic materials in the aquifer, in large part because magnetic materials are relatively easy to measure. We also want to determine the amount of magnetite in aquifers because of the association of magnetite with magnetic susceptibility. And we also want to identify the mechanisms for abiotic degradation in both aerobic and anaerobic water supply aquifers. So in order to get this project underway, we started by picking 5 sites to collect soil and groundwater samples from. Those sites are a former manufacturing facility in Arizona, the former Twin Cities Army ammunition plant in Minnesota. There's actually 2 locations there where we collected samples from, Site 102 and Site A. Site A is where degradation of cis-DCE has been detected, but no PCE or TCE. Vandenberg Air Force Base in California and Plattsburgh Air Force Base in New York. From each of those sites, we've already collected about 70 kilograms of soil cores and then groundwater from nearby wells. The soil samples were delivered to my lab at Clemson, where they were dried and homogenized. We then sieved them into 2 fractions, lower and above 850 microns. And then the sample below 850 microns, we've separated into magnetically enriched and magnetically depleted materials. The driver behind this is the hypothesis that there's an association between magnetic susceptibility, which is a measure of how much magnetic material is present in a sample, and the rate constant that has been observed in microcosms and field studies for first order degradation rates, abiotic degradation rates. What we need to pay attention to on this slide is the range of magnetic susceptibilities that's measured in meters cubed per kilogram. And the range extends over about 2 orders of magnitude, from 10 to the minus 7th to 10 to the minus 5th meters cubed per kilogram. And there's been a general association between magnetic susceptibility in that range and increases in the abiotic rate constants. So we set out to find sites, 5 sites that have a range of magnetic susceptibility, so we could then measure the rate constants and further evaluate if it's the magnetic component of those soils that is a significant driver in the abiotic degradation. As shown in this slide, this covers data from microcosms for several of the chlorinated Ethenes, as well as data from field sites and some laboratory studies. So as I mentioned, the soil was delivered to Clemson University, where we homogenized it and dried it. We considered grinding it, but opted against doing that out of concern that grinding might alter the surfaces where the minerals are located. We did, however, then sieve the dried samples into 3 size fractions, and then we separated those fractions that were less than 850 microns into magnetically enriched and depleted using a strong bar magnet. We actually intended initially to use something called a France separator, but found that the bar magnets was just as effective and even more so. So these 2 pictures in the insets show a sample that's been magnetically enriched. You can see the collection of the magnetic particles on the bar magnets. We collect that and then scrape it off to get the enriched fraction. And then in the depleted fraction, you can see there's very few particles that collect there. Once we've separated into the fractions, we calculate the degree of enrichment based on the magnetic susceptibility. So this shows that particular metric, measuring the magnetic susceptibility of each of the fractions. You can see on the bar graph that we measured magnetic susceptibility and then converted that into the magnetic susceptibility is in units of SI. You then apply the volume of the measurement cell and the weight of the sample to get it in units of meters cubed per kilogram. Then use this equation to convert it into magnetic material, followed by this equation that converts it into a percent. So we took the sample from the Arizona site and measured the percent magnetic material in the sediment as received, and then in the different size fractions. And what you see here is that the magnetic component increases with the smaller size fraction. So for this project, if under ideal situation, we would have picked the less than 150 micron fraction to do the experiments with. But I don't show on this slide the weight fraction, and the smallest fraction was the lowest amount of weight. So we wouldn't have enough material if we used just that smaller fraction. So we ended up deciding to use the full 850 micron material, and I'll show you in a minute that gave us adequate enrichment in magnetic material. So that's what this slide shows with respect to magnetic susceptibility. The units on the y-axis are meters cubed per kilogram. And as I just recalling your attention to that earlier slide, we're looking for activities in the range of 10 to the minus 7th to 10 to the minus 5th. So these are the 5 sites: Phoenix, TCAP-A, TCAP-102, Vandenberg, and Plattsburgh. And what you can see is that with TCAP-A, TCAP-102, and Plattsburgh, there is a very high level of magnetic susceptibility in the enriched fractions. In all cases, as expected, the depleted fraction shows little to no magnetic susceptibility. And then there's a variety of magnetic susceptibilities in the unaltered fractions, meaning the fraction that we didn't apply the magnet to. The point of this slide is to communicate that we were successful in identifying sites with a range of magnetic susceptibilities, so we could test the hypothesis that magnetic susceptibility is related to the abiotic degradation of the chlorinated Ethenes. When we express that magnetic material as a ratio of what is in the enriched fraction on the y-axis, the magnetic material in the enriched fraction divided by the magnetic material in the unaltered fraction, you can see that range of activities. Now, the interesting thing is that in the samples, Vandenberg is a big site. In the particular sample that we got from that site, there was very little magnetic material in it. And so at the moment, that sample is in effect going to serve as a negative control for us. The Phoenix sample, which I'll show you in a minute, the C14 results for a modest level of enrichment in magnetic material. It's really quite high for TCAP-A, 102, and Plattsburgh. When you express that ratio of enriched to the depleted fraction, so the left side is just to the unaltered fraction, this panel is for the depleted fraction, you can see that ratio is really enormous for TCAP 102, quite high for Plattsburgh, and also TCAP-A. It's still notable for the Arizona site and really not detectable for Vandenberg. So we got this nice range of variations in magnetic material among the 5 sites. Okay, so we're using this now in microcosms to measure the rates at which the chlorinated Ethenes undergo abiotic degradation. We use 160 ml serum bottles with soil, and to that we add groundwater, but we filter sterilize the groundwater to make sure that there are no microbes present. We're doing these microcosms with the original wet soil as received, with dry sieved soil that we haven't done any magnetic processing to it, another set with the magnetically enriched soil, another set with the magnetically depleted soil, and then a set of controls with just the water in it to measure the background rate of C14 product formation, which we subtract away from the activity in the treatments with the soil present. For each of the soil types, we have 3 treatments. One is anaerobic, prepared in a glove box, and then we sparge it to get rid of the hydrogen in the headspace. We've got another set that's anaerobic, and we added a hydroxyl radical scavenger. The hypothesis there is that the abiotic degradation we're seeing is attributable to a hydroxyl radical, and by adding the scavenger, we're testing for that. And then another set for aerobic, because a lot of these water supply aquifers are aerobic, and we want to see if abiotic degradation is occurring in those as well. The preparation of the microcosms, as I said, 160 ml serum bottles, pretty standard stuff there. Teflon septa, 100 grams of wet soil, 70 grams of dry soil, 70 mils of filter sterilized groundwater. Once we've got it prepared, we inject into the headspace of the bottle the carbon-14 labeled materials. And then we also add some unlabeled TCE and PCE to get the initial concentration in the vicinity what's applicable to a lot of these sites where there's persistent contamination below about 1 milligram per liter. And then we incubate the bottles on a tumbler and remove them to make weekly measurements of the polar products that form in 5 ml samples that we filter to get out, take away any possibility of measuring adsorbed material. And then we analyze how fast the products form to determine the abiotic degradation rate constant. The way that this is a key component of the whole assay is that to the bottles, we add purified carbon-14 labeled TCE and PCE, and we're going to extend that to cis-DCE. We get the labeled material from a vendor in California, Moravec Biochemicals. We prepare a stock solution of that, and then we check its purity. And so far, we've been very happy with that being over 99%. The way that the purification is done is we inject the stock solution with the labeled compound in it onto a gas chromatograph. The gas carrier, nitrogen gas, sweeps the stock solution through the column. It then, as it elutes from the column, we bypass the detector and have it exit the gas chromatograph. And at the time when the chlorinated labeled compound elutes from the column, we inject that into the headspace of the serum bottle to get the carbon-14 labeled material into the bottle. So that's a key component of this. For vinyl chloride, we're going to do that by, we can't get a source of labeled material, so we're going to make it in the lab using a culture that at low pH stops reductive dechlorination at vinyl chloride, and then we'll inject that biologically produced vinyl chloride. Once we have the serum bottles assembled, they have to be incubated on a tumbler. We created our own tumbler using a 55-gallon drum, and in that we put 7 layers of serum bottles, and once it's all filled up, we lay it down on industrial rollers, which we can tumble at a low rate to keep things gently mixed. And then when it's time to sample, we open up the drum and take the bottles out and remove the 5 ml sample. Okay, so this is an example result that we have so far from the Phoenix site and the original wet sediment. And this panel on the left is for the aerobic treatment. The y-axis on both of these graphs is in units of disintegrations per minute per mil. So the way that you measure C14 products is in an instrument called a liquid scintillation counter, and that reads out in units of DPM, and that's what we're plotting here. So, and then we run the assay over a 6-week period, 42 days. We found that that is adequate for us to establish a robust rate. And so the panel on the left shows the results for triplicate bottles. All of the treatments are done in triplicate. And this was a situation where, again, it was the original wet sediment under aerobic conditions, which actually that particular aquifer that we got the sample from does have enough DO in it to categorize it as aerobic. And what you can see in here over time is a steady accumulation of 14C labeled products from the 14C labeled TCE that are accumulating over time. The panel on the right shows the same treatment, but without the soil present. So that has just the filter sterilized groundwater. What we're checking for with these controls is to make sure that there's nothing in the filter sterilized groundwater that is reacting with the TCE and yielding products. We want just the products associated with reaction of the TCE with the soil. And so we use the control data to adjust the experimental bottles to come up with a net rate constant. And that's what I'm going to show you on this next slide. By sampling the bottles on a weekly basis and measuring the products, we can determine the first order rate constants. This graph shows the results for the wet soil, the soil that's been dried and enriched in magnetic material, the sediment without any enrichment, the magnetically depleted soil, and then the controls with no sediment. And then here are the 3 treatments: the anaerobic treatment without a scavenger, the anaerobic treatment with the hydroxyl radical scavenger. And I just discovered that was a typo. This is the aerobic treatment with no scavenger present. Okay, so what we find here is that the highest first order rate constant for this particular site with TCE was in the original wet sediment. So obviously drying did tamp down the activity a little bit. That means the wet sediment's the most relevant to actual field conditions anyway. There were some minor differences for this site between the magnetically enriched and the unaltered sediment, because at this particular site, the degree of magnetic materials is on the modest side. However, the magnetically depleted sediment was definitely lower in the anaerobic and aerobic treatments without the scavenger present. The scavenger results were lower, and that clearly suggests that there is a possible role for the hydroxyl radical and the abiotic degradation of TCE. Now, these, what I'm calling raw first order rate constants, need to be adjusted because they're not reflecting the ratio of water to soil that's in the bottle. And that's actually a moving target because we're removing water samples over time during the incubation and changing the water to soil ratio over time. So we've come up with a way to translate those raw K values into soil normalized K values, which I'm going to show you on this next slide. So the upper right panel, the y-axis is the first order rate constant, but this time rather than just per time units, it's liters per gram per day. So we've normalized it to the volume of water and the mass of soil that's present in the bottles. And then in the below that, we show a first order rate constant that's relevant to the aquifer. Okay, and the same treatments: anaerobic without a scavenger, anaerobic with a scavenger, and aerobic without the scavenger. So what these results show is that the normalized first order rate constant to the amount of soil and groundwater in the bottles. It corrected for, well, the filter sterilized groundwater corrects for the activity in just the water. And so basically these normalized values follow the same trends that we saw in the previous slide for the raw K values. Now, once you have a first order rate constant from the microcosms, that can be converted into a first order rate constant for the aquifer using the ratio of soil mass to groundwater that's found in the aquifer. That's what we did in this lower bar graph. And from this, the important take home is that in the aerobic aquifer with the wet soil, this purple bar right here, that rate constant translates into a half-life of about 1 year, which is certainly a meaningful outcome for assessing the potential for attenuation. All right, so just to bring this home with some key points, we collected soil samples from 5 sites that display a wide range of magnetic susceptibility and percent magnetic materials. We've gotten the 14C assay to work and refined to factor in the amount of soil and changing amount of groundwater in the microcosms during the incubation so that we can then relate what's happening in the microcosms to an actual aquifer. At the Phoenix site, we saw the highest rate constants in the with TCE in the assay for the original wet sediment, which, as I indicated, is the most relevant to in situ conditions. It turns out there that the aerobic treatment has a rate constant in the rate constants with the dry soil increase as the magnetic susceptibility increases, providing us with some initial insight into the idea that as you see an increase in magnetic susceptibility, you'll see an increase in the rate constant. As I just mentioned, the aquifer half-life for the aerobic wet soil was about 1 year. And what I'm not showing here, but we now have results for the same site with carbon-14 labeled PCE. And in all cases, the results with PCE are lower than, indicate lower reactivity than the treatments with TCE. So what does this all mean to DoD? I think the tools that we've developed with the C14 assay are going to give DoD remedial project managers a way to help assess site-specific degradation rate constants. The assay takes about 2 months and it's affordable. The results can be used to improve site conceptual models, especially if you have a hypothesis that abiotic processes are contributing to attenuation. They're going to allow you to include abiotic degradation in remedial timeframe estimates, and the scientific evidence and provide support to bolster the credibility with regulators and stakeholders. I think anytime that you can provide evidence to regulators and stakeholders for degradation occurring based on product formation, you have a pretty solid case to make. Going forward, we got to apply the assay to cis TCE and vinyl chloride. We're on the cusp of doing that. We need to further refine that assay for the soil normalized rate constant, so we have error bars associated with them. We're working on that because anytime you have a rate constant, you certainly want to know what the error bounds are on that. We also want to evaluate laboratory synthesized magnetite as well as iron 2 adsorbed to the magnetite to test out that association between magnetite and abiotic degradation. And then we've a whole nother part of the project is to look at the enrichment in carbon-13 and 37Cl in microcosms with all 6 of the chlorinated Ethenes without C14. So we can learn some new information associated with carbon-specific isotope analysis. And then we also want to assess the overall importance of magnetic materials to rate constants and pathways, so we can further advance the field in understanding the contribution of abiotic degradation. All right, and with that, I just want to acknowledge my collaborators on this, Alex Poole and Alex Ramos Garcia at Clemson University, although Alex Ramos Garcia just recently went to ERM. John Wilson at Scissortail Environmental Solutions. John is really the brainchild behind this idea and has been the leader among many of us in promoting the applicability of abiotic degradation as a contributor to remediation. And then Thomas Kuder at the University of Oklahoma, he'd be leading the effort to do the compound-specific isotope analysis. And got some references on this slide and a place for you to find additional information. And at this point, I would be happy to answer any questions if you have them. Thanks for listening.
Fantastic. Thank you so much, David. And to our audience, I'd just like to remind you at this time that you can submit your questions using the Q&A box through the Zoom platform. And we do have a number of questions. David, we're going to start with just one or two clarification questions before moving on to others. Can you clarify what exactly was measured in the liquid? Did you measure chemical species in the liquid phase? And did you measure 14CO2 in the headspace?
Okay, so in the assay, what we do is take a 5 ml sample from the serum bottle. We then filter that to remove any particles so that there's no activity associated with absorbed TCE to the particles. And then we subject that to both acidic and alkaline sparging. What that allows us to do is to actually quantify how much of the products formed are in the form of carbon dioxide, which is going to be in the alkaline fraction, and then soluble polar products, usually in the form of organic acids that remain both in the acidic and the alkaline fraction. In this study, we have not targeted analysis of those specific polar products that might be present. We've done that in previous studies. So we have an opportunity to do that, but our focus mainly is on total products at this point.
Okay, thank you. Given that many of the abiotic products can be volatile, like ethane and propane, can you comment on whether you're missing any activity or what the products might be?
So that's an important question. In this form of the assay, with the C14 assay, because we're sparging, we are not quantifying the formation of the gases, acetylene, ethene, and ethane, and then also some other volatile products that could form like propane. We're not quantifying those. We will be doing that in the microcosms that we prepare for carbon-specific isotope analysis. We're not going to be altering the headspace, not going to be sparging the headspace of those bottles. So we'll be sampling those over time and capturing the gas formation. And then that will position us to combine the rate constants that we measure from those gases with the rate constant from the CO2 and polar products to get a total rate constant.
Okay, great, thank you. Can you share your thoughts on what might be generating hydroxyl radicals in the system?
That's honestly an interesting question. The short answer is we don't know yet. If hydroxyl radicals were at the center of the activity, one would expect that the greatest amount of activity that we would see might be in the aerobic bottles. But in fact, it's the anaerobic ones, at least with the Phoenix, that we've seen higher amounts of activity. So you would have to expect that there's something involved with oxygen. The anaerobic ones are set up in a glove box, and we work hard to keep oxygen out, but it's not to say that a small amount may diffuse into the serum bottles that contribute to the formation of those radicals. But we're hoping actually that the results from the C13 work and the 37Cl work will give us some of the mechanistic answers that will give us insight into how the hydroxyl, how the free radical may be operating.
Great, and following on that, why would the anaerobic treatment from an aerobic aquifer have the higher rate?
Yeah, that one thing that we are intending to look at is the possibility that there are other free radicals that may be present, sulfur-free radicals or iron-free radicals that may be the ones operative in these microcosms.
Do you think that there's any other mineral that might be more important than magnetite? How would that affect your interpretation? Something else we're also contributing to the degradation.
Well, that's the beauty, if you will, of doing the assay. We're not, the results aren't dependent on knowing which mineral is active. The hypothesis is that magnetic materials are a significant contributor, and the earliest results we've had seem to be consistent with that. But it's not to say that there are not other minerals that are present in the soils that can be contributing to abiotic activity. And when you do an observational assay, like with C14, in effect, it doesn't matter which mineral's doing the work. The question is the abiotic degradation occurring? And we can uncover that.
Excellent. And what about aerobic co-metabolism? Is there the potential that under aerobic conditions that co-metabolism is contributing to some activity in the aerobic wet samples?
Well, so you have to first remember that the water that we're using in the assay has been filter sterilized. So we know that there's no contribution of biotic activity there. I think this comment has come up in the past about the possibility that there's some microbial activity remaining in the soil. I tend to think that the chances of that are diminishingly small because the soil's been dried and stored under dry conditions for many months before we put it into the serum bottles. But to test whether or not there's any contribution from biotic from those dried and sieved samples, we are going to run some controls with acetylene as an inhibitor, and we may also spike some of the samples with live bacteria and then run them through the drying and sieving process to see if they survive. So we are going to explore that option to rule out the contribution of any biotic activity in these microcosm assays.
Great. Then zooming out just a little bit, we have just a couple more questions for you in this round. Given your results, do you think it might be a strategy to add magnetite to sites to enhance abiotic degradation?
I don't see that as a way to go. We're really oriented towards sites where there have been low levels of contamination, in the part per billion range for many years, sites where managers have relied on pump and treat for decades as the remedial strategy. And the elephant in the room is, can you turn those pumps off and then achieve a level of degradation activity where abiotic processes will contribute a lot to them. So I think that's where we're headed with this whole effort, is to give site managers a tool to help them assess whether or not abiotic is a viable natural attenuation remedy. If you were going to go towards an active remedy and you wanted to use iron-based materials, there are things like much more reactive nanoscale type iron that can be injected as one approach. But active remedies are going to cost you a whole lot more than what we're aiming for, which is a situation that you can demonstrate activity to regulators that natural attenuation is sufficient to keep the contaminants on your site, or possibly to make sure that the contaminants are going to reach cleanup goals.
And David, is this 14C assay available for use at other sites beyond this SERDP project?
Yes, it is. We are partnering with Microbial Insights to make the assay available, and we have one pretty good success story with a client who did the assay and then was able to use the information from the assay to go to regulators and say, the rate constants that were observed in the laboratory study are consistent with the rate constants that we're observing in monitoring wells, and the assay is providing evidence that degradation products are forming. And that was central to their ability to reach a natural attenuation remedy. So yes, through a partnership with Microbial Insights, we make the assay available to others.
Great. Thank you so much, David. And David will come back at the end of the webinar for some more general questions for both of our speakers. Thanks again. And it's now my pleasure to introduce our second speaker, Dr. Demin Fan, who is an environmental scientist at Geosyntec in San Diego, California. Demin's areas of research focus on developing and characterizing abiotic iron-based remediation technologies for active and passive management of plumes and the use of carbonaceous materials for the in situ remediation of organics. He has served as the principal investigator or key performer for several research projects on quantitative characterizations of natural attenuation processes. He's authored more than 20 peer-reviewed research papers, including on novel tools to characterize natural attenuation processes. He earned a bachelor's degree in environmental chemistry from Nanjing University in China, a master's degree from University of Delaware, and a doctoral degree in environmental science and engineering from Oregon Health and Science University. Demin, please proceed.
Thank you, Jennifer, for the introduction. Thank you for the opportunity to present this research funded by SERDP. So David's presentation set up my presentation nicely. So the project I'm going to talk about today is essentially addressing the same line of problem, which is the challenge we as practitioners are facing to quantitatively characterize abiotic natural attenuation rate. But we're looking at the problem from sort of different perspective. Instead of looking at contaminant degradation rate, which David did, we're trying to focus on characterizing the reactivity of the reactive minerals that are responsible for the abiotic contaminant degradation. So before I start, I want to acknowledge my collaborators, Dr. Paul Tretnick and Rick Johnson from Oregon Health and Science University, and Dr. Chris Conker from Royal Military College of Canada. So I will start with some project motivation, focusing on why we need better tools to characterize abiotic natural attenuation. Now I'll walk you through sort of key stages of the R&D process, all funded by SERDP, evolving from proof of concept in laboratory to develop a field-deployable ORP kit that can be used in practice. The last bullet is really the scope of the ongoing our SERDP ongoing projects. By walking you through the process, I hope you help you better understand where we're coming from and where we're going forward. So I'll conclude with some discussion on next steps and benefits to DoD. So the major motivation for this project is that abiotic natural attenuation rates of coronary Ethenes in particular are usually very slow and difficult to quantify in practice. So David just gave a nice presentation on C14 acid potentially can shorten the time frame of laboratory evaluation to 2 months. But for a conventional microcosm study, it is very difficult to convince site managers, as many of you are, to fund a multi-year microcosm study that in the end may only produce 5% of concentration reduction. Even that 5% concentration reduction could potentially grow to 10%, 20% over a longer period of time, which might be relevant to abiotic natural attenuation. So what we're trying to do here is trying to develop a better tool to help you more quickly and more efficiently identify and characterize the reactivity of those reactive minerals. Additionally, I want to mention some of the recent work led by Paul and Rick at OHSU and also Michelle at Iowa and others, which show that many of those reactive minerals are actually metastable intermediates that undergo complex transformation driven by microbial redox processes. So the existing tools we have are mostly static. You take samples from the ground and do a bunch of characterization, and those tools will not be able to capture those kind of transformation process. So this slide summarizes the existing tools we often rely on to characterize ANA, which can be grouped into 2 categories. On the left is a reactant, on the right is a contaminant. So on the reactant side, commonly people use aqueous geochemical indicators such as ferrous iron or oxidation reduction potential or dissolved oxygen to qualitatively assess redox conditions. But as we know, aqueous geochemistry hardly equals to solid phase reactivity. I'll give you a very good example in the next slides. We have also used aquifer solid mineralogy, such as X-ray diffraction or electron microscopy, to identify and differentiate those reactive phases. But again, they do not speak to the reactivity. Magnetic susceptibility, you heard from the previous presentation, has been used, measured in situ and used to correlate with decoordination rate, but they might not be sensitive to non-magnetic minerals that are reactive, such as iron sulfide. So on the contaminant side, people have used sort of parallel decreasing trend in both parent and daughter products as an empirical evidence to demonstrate the occurrence of abiotic natural attenuation in the field. But the concentration pattern can be pretty easily distorted by site hydrogeology and heterogeneity. So this slide gives you a good example that aqueous geochemistry does not equal to a solid phase reactivity. So the purple solution on the left first figure is a residuum, which is a redox probe. When it gets reduced, it will change the color to pink. So here we attach that residual solution to a column that is reducing. In the second picture, we pull the pool water out from the column and mix with the residuum. You can see the purple becomes lighter, but the color did not change. In the third picture, we re-injected this solution back into the column and pour out the water again. So you can see the color now changed to pink. So that's a perfect demonstration showing that majority of the reducing capacity that reduce the redox probe from purple to pink is actually associated with solid phase present in that column. So now the question becomes, can we find a sort of universal descriptor that can quantify the reactivity of that solid phase and further correlate that descriptor with abiotic contaminant degradation rates? So we found some good examples in the literature. These two figures are from Chris Gorski and Michelle Sherrill's early work on magnetite. So in this study, they synthesized the magnetite with different iron 2 to iron 3 ratio and reacted with nitrobenzene. So it shows both nitrobenzene reduction rate constant, which on the left, and magnetite reduction potential on the right had linear correlations with iron 2 to iron 3 ratio. This suggests that reduction rate constant of nitrobenzene and mineral reduction potential also form a linear correlation. So this indicates that reduction potential of the minerals can potentially serve as a useful indicator for abiotic contaminant degradation. But in this study, the mineral reduction potential was actually measured by packed electrodes, which was customized and may not be widely accessible for broader uses. So one of the easy way and one of the most obvious way to measure reduction potential that everyone in this field knows about is to use an ORP electrode. But conventional ORP measurements is only sort of suitable for aqueous phase measurements. The electrode has inherent limitation to see the solid mineral phases because there lack of direct communication between the mineral surface and the electrode surface. So while the reactive minerals can be a strong reductant, the exchange current density on the surface of the electrode could be quite low. So essentially the electrode is blind to the reactive minerals. So how can we overcome this challenge? One way to do that is to add Electron Shuttle into the system. So what is Electron Shuttle? Electron Shuttle has rapid redox reversibility. What it does is it rapidly equilibrates with reactive minerals. So it has the same reduction potential as the minerals. The electrode will have no problem measuring the Electron Shuttle because it is soluble. But essentially, the Electron Shuttle serves as an interpreter to translate the mineral reduction potential to the electrode. So in order for the Electron Shuttle to work, its potential range also needs to match with the mineral reduction potential. So this is a very important concept I want you to remember. So I'll come back to this several times later throughout the presentation. So the last question is, how do we know when we measure the accurate mineral reduction potential? Fortunately, many of the Electron Shuttles also have colors, and their color changes upon oxidation and reduction reaction. So we can actually use the colorimetric approach to measure reduction potential and compare that with electron measurements. So as I mentioned at the very beginning, the work is developed in 3 key sort of SERDP projects. There are also many other SERDP and NSF projects support the research development of using Electron Shuttle to measure the ORP for assessment of abiotic natural attenuation. So the proof of concept of this idea really came out of the ER 2308, where we used individual Electron Shuttle to measure ORP of model minerals, but mostly using the colorimetric approach. So we did limited amount of the electron measurement towards the end of this project, which was really promising. That led to the second phase, second SERDP project, where we transitioned from the model minerals to actual aquifer solids collected from the DoD sites using electron measurements with individual Electron Shuttle. So these two projects collectively led to our ongoing SERDP projects, where we aim to develop a field-like kit using a mixture of Electron Shuttle instead of individual Electron Shuttle. So that's the key difference between our ongoing SERDP projects, the previous two SERDP projects. So I'll explain why we're doing that later. So in the next part of the presentation, I'm going to highlight some of the key findings from each of the three stages. So during the proof of concept, we use iron 2 doped synthetic iron oxides as our model reductants. We added a small amount of individual Electron Shuttle to mineral suspension. Then we measure absorbance spectra of the Electron Shuttle in the aqueous phase, after filtering the suspension, which is shown on the left. So based on absorbance spectra, you can calculate how much Electron Shuttle was reduced by iron oxides, and you can use this Nernst equation to calculate the reduction potential of the Electron Shuttles, which equals to the reduction potential of the minerals. So on the left, we basically use electrode to measure the same mineral reduction potential in the presence, initially in the absence of Electron Shuttle. Then we added Electron Shuttle into the system to see how the potential responds to Electron Shuttle addition. So this example results showing here, you can see in some cases we see a very nice potential decrease in response to the addition of Electron Shuttle, but in other cases where we see no changes. So this goes back to the earlier point I want to make is, in order for Electron Shuttle to work in this kind of system, the Electron Shuttle needs to have a matching reduction potential as the minerals. So during this process, we did a lot of colorimetric measurements because we're pretty confident that this is the accurate measurements, and we use those results to validate the electron measurement results. So this graph shows the comparison between the two measurements. The x-axis is the reduction potential determined by the colorimetric approach, and the y-axis is the potential determined by the electrode. So the red open symbols are the measurement by the electrode without Electron Shuttle, and the blue dots are the measurement by electrode with Electron Shuttle. So if the two approach produce consistent results, you would expect that the points fall onto this one-to-one diagonal line. So you can see for all the blue dots, they are showing more negative reduction potential than open red symbols. And also they're falling much closer to this one-to-one diagonal line. This definitely suggests that Electron Shuttle improves electron response to the reactive minerals. So then we want to know if the mineral reduction potential can be correlated with contaminant degradation rate, which is the ultimate goal of this approach. So we selected three different contaminants, three different classes of contaminants, and here, and measure the degradation kinetics of them by the minerals. So in the figure on the right, to the right, we basically plotted the surface area normalized rate constant versus the mineral reduction potential. Here we're determined by the colorimetric approach. So you can see, despite a couple of outliers here and there, overall the linear correlation exists for all three different classes of contaminants. This is really encouraging because if with the linear correlation between the rate constant and reduction potential are universally applied for different types of contaminants, we can anticipate there's a linear correlation for coronary Ethenes as well. So the proof of concept shows Electron Shuttle could improve the electron response to reactive solids, and the measured mineral reduction potential can also be correlated with contaminant degradation kinetics. Then the next step we're trying to do is we're trying to see if these two findings can also be sort of replicated with the contaminant degradation, with aquifer solids collected from actual contaminant sites. So through our collaboration with an ESTCP project, we were able to get some aquifer solids from 4 DoD sites where ZBI remedy was implemented in the past. So in this case, we expect ZBI has been transformed to various forms of iron minerals in the field. Therefore, these aquifer solids provide iron-rich materials that are ideal for assessing abiotic natural attenuation process. So the materials were collected from 9 different locations, including treatment area, background area, and downgradient area. The materials were collected by cryogenic coring method to preserve the biogeochemical conditions of the cores. So the frozen cores were shipped to the lab and processed in a way to minimize exposure to oxygen. So we measured the reduction potential of aquifer solids in this stage only by the electron measurements, because the electron measurements are already validated with a colorimetric approach during the proof of concept, and electron measurements definitely represents a more field-friendly approach. So this figure shows the potential reading as a function of the 3 different solids. You can see after adding electron shuttles, the potential become more negative, replicating what we saw with the model minerals. So this indicates that we observed improved electron response to aquifer solids due to the addition of electron shuttle. So one thing I want to point out here, for 3 different solids, we actually using 3 different electron shuttles. This is because, as I mentioned earlier, so going back to the point I made earlier, in order for the electron shuttle to work for this purpose, the mineral reduction potential needs to be within the potential window of the electron shuttle, which is usually plus or minus 50 millivolts of the standard reduction potential of the electron shuttle. So this is a major limitation of this method, and it is also the motivation for our ongoing SERDP project, which I will discuss later. So we also measured reduction kinetics of carbon tetrachloride by aquifer solids. Here we use carbon tetrachloride as our probe contaminants. So the experiments were conducted in this type of rotating the tube using high solid to water ratio to simulate actual porous medium condition. The example degradation kinetic is shown in this figure. So we can see nice first order reaction pattern for 4 different solids collected from 4 different areas. Then we wanted to know if the sort of mass normalized first order reaction constants is correlating with the mineral reduction potential. You can see the linear correlation is pretty tight. It's pretty promising given the material actually coming from different sites and different areas. They are well bounded by this 95% confidence interval. So this is very encouraging because we know the idea of correlating the thermodynamic and the kinetic aspects of ANA evaluation works with real size samples, and this tool has a potential for practical applications. So also we would like to test if a simple commercial ORP electrode could produce similar potential readings as a well-controlled laboratory potentiostat. So if that's the case, the equipment requirement for this measurement will be much less, and the method will also have the potential to be carried out in the field. So this figure shows the comparison between the 2 approaches. So we see fairly consistent results from between the commercial ORP electrode and the laboratory well-controlled rotating disc electrode. We see some kind of deviation above the 0 millivolts. This is mainly because we do not have a suitable electron shuttle that measure above the 0 millivolts. So this really motivates the ongoing SERDP project, which is to develop a field kit and a protocol for ORP measurements. So earlier I mentioned in order for electron shuttle to work, the mineral reduction potential needs to fall into the potential window of the electron shuttle. So this represents a dilemma. So you have to know the mineral reduction potential in order to select the right electron shuttle to measure the mineral reduction potential. So in the field, you don't likely have the luxury to test a different electron shuttle, see which one works with your solids. So what we propose here is to test a mixture of electron shuttle, that covers a broader range of reduction potential. But the idea is the aquifer solids will automatically respond to a suitable electron shuttle within that cocktail, while the other electron shuttles will not impact the system. So this will greatly increase the practicality of ORP measurements in the field. So in this project, there are 4 different tasks. First, we want to validate the kit concept. Here we're going back to using the model minerals. It sounds like a backwards, but we want to make sure that there's no unexpected things happening when we actually mix everything together as a cocktail. And secondly, we want to develop a field protocol that allow us to measure this in the field. So field measurements needs to be a simple, a quick, and efficient approach. Thirdly, we want to optimize the field protocol and ORP kit with aquifer solids have already been tested. Finally, we want to test the effects of different operational variables to see which are important and which are not. So to validate the kit concept, we used model minerals under well-controlled conditions. Here, we're using the anaerobic chamber and well-mixed reactor. The figure on the left shows the ORP as a function of time. Over time, we're adding the electron shuttle individually from more positive reduction potential to more negative reduction potential. So on the right, in this case, we're actually adding a mixed electron shuttle solution at individual stages with the 3 different doses from low 1 micromolar to high 80 micromolar. So the pink curve is the response from the glossy carbon electrode. The purple is the platinum electrode, and the green is the pH of the reactor. So on the left, you can see the ORP dropped twice in response to the first 2 addition of electron shuttles. On the right, the ORP also responded to the cocktail addition. So in both cases, the ORP plateau reached a similar value, so indicating that the cocktail concept actually worked under the tested conditions. Then we want to see if the same measurement can be achieved using a more field-friendly procedure. Here we're still using the model minerals, but switching from anaerobic glove box to a portable glove bag. So we recognize a glove bag is still not very ideal for field measurements, but at least it is a workable solution if the anaerobic condition needs to be maintained in the field. This picture just showing the experimental setup. So we have a 40 ml bottle vial with a multi-hole cap that housing the electrode and nitrogen and gas line. And when we add a mixture of electron shuttle into the reactor, then the ORP will be continually recorded outside the glove bag. Here's some results, still quite preliminary results. Here we're using the 0.5 gram per liter Gertai with a different concentration of iron 2. We're seeing the general trend of ORP response to the addition of mixture electron shuttle is consistent with the previous findings. For example, here we have a 20 micromolar addition. We're seeing a nice job of ORP in response to that addition. And when we add 100 micromolar, it's also show a good job. But the problem we identify here is the absolute ORP value of this system is different from the ORP measured in the well-controlled system in the anaerobic chamber by nearly 200 millivolts. So we're still in the process of diagnosing the cause for this deviation. We think it's probably caused by some of the residual oxygen present in the glove bag that was not completely removed during the evacuation process. So in terms of the next step, we're going to continue developing our field protocol, looking at different operational variables that we can potentially encounter in the field, such as the oxygen level. Also, we're interested in looking at the effects of non-reactive solids. Typical aquifer solids only have a few percentage of the reactive phases. Majority of the mineral phase might not be that reactive. We want to see how those fractions will impact the ORP measurements. Finally, we also want to look at the effects of mixing on the ORP measurements. Then we want to test the optimized field protocol with aquifer solids that previously characterized. Also want to coordinate it with other projects to make measurements with site materials of interest for ANA assessments. So to conclude, we show that electron shuttle results in a more accurate ORP measurements for the reactive solids. So we advanced the ORP measurement from only be able to measure the aqueous phase and to be able to measure the reactive solids. The electron shuttle mediated ORP values of reactive solids can also be correlated with contaminant degradation kinetics. We demonstrate that works for different types of contaminants. So the last one is sort of, we're still in the work in progress to develop these tools that can be potentially useful for field measurements. So benefits to DoD, the tool we're developing here is trying to add a more qualitative tool into our existing toolbox to help understand the reactivity of these reactive minerals. So as I mentioned earlier, most of our existing tools for reactive minerals are qualitative and do not really speak to the reactivity. The ability to measure the reduction potential of minerals, which sort of dictates the thermodynamic driving force of contaminant degradation reaction, will significantly improve that aspects. And by doing so, we hope the tool could help better integrate ANA aspect into the existing M&A evaluation framework, which heavily focused on the biological M&A process. So here are some references. Additional information link. So with that, I would like to thank you for your time and happy to answer questions.
Great. Thank you so much. And we do have a few really great questions to everyone who's listening. Please ask other questions in the Q&A box. So first, Damin, is there a rule of thumb ORP value above or below which abiotic natural attenuation might be significant?
That's a good question. So I think everybody wants the number, it's magic number. At this stage, I think if we go back to one of the slides I showed you earlier. So let's see which slide number here. Sorry, I don't have a slide number in front of me, but anyway, so what we showed that the correlation between the reduction potential and the carbon tetrachloride degradation kinetic, I think the measured mineral reduction potential has a range from minus 200 millivolts all the way to 300 millivolts versus standard hydrogen electrode potential. So then correspondingly on the rate constant, we have also 5 or 6 orders of magnitude difference on the y-axis. So I think it's really hard right now to pinpoint a specific potential number that can be used to decide, above which number we're going to see or not see abiotic reduction potential. What we hope to do is we can sort of transition from the carbon tetrachloride measurements to get a more relevant reduction rate constant measured for TCE. Then we can see construct another correlation where we measure the reduction rate constant for TCE and also correlate with the mineral reduction potential. So if that correlation works, I think we should be able to provide more information on the potential range. Here we go. That's a slide. Yeah, you can see also on the right-hand side we have this half-life laid out from days to centuries. But this works for the carbon tetrachloride. So I think for TCE or chlorinated, we would expect that the straight line moved down by a lot. If we have that correlation established, then we can be more confident saying pinpoint a potential range where we think the TCE or chlorinated abiotic dechlorination might be more significant.
Wonderful. Thank you. I like how you laid out those half-lives on the right-hand side as well. In the case, Damin, when you were showing the mixture of the electron shuttles, when you do that, do you get any information about which one is actually the most suitable one? Like, how can you tell which electron shuttle is really interacting with the solid?
Yeah, well, that's sort of the whole premise of this approach is the solid phase will sort of automatically respond to the right partner of the electron shuttle. So in that sort of the earlier two SERDP project, we kind of tested individual electron shuttle. We know the standard reduction potential of those redox probes. Then we apply those in the in our sort of system, then we know when the mineral responded and when the mineral does not respond to the addition. So we kind of already paired the minerals with the right electron shuttle. So we kind of assume in the mixture that mineral will basically respond to the same electron shuttle that we identified in the previous 2 stages are suitable electron shuttle to that minerals. Don't know if that's answered the question perfectly.
Yeah, I think there's a way for it, it sounds like. So we have a number of questions about the field application of this approach. Can you tell us a little bit more about the sampling requirements for an eventual field kit? Will you have to collect cores and maintain anaerobic cores prior to the measurement?
Yeah, that's a good question. Our sort of current idea, we haven't really actually gone out to test the field protocol with the core. So there's a number of variables we have to work out in lab first. For example, one is anaerobic conditions. I think in our recent experiment, we definitely see some interference from the atmospheric oxygen. So in the second stage where we do the cryogenic approach, there's some people asked a question about earlier before about whether it's needed, the cryogenic approach is needed. So we're still trying to explore that. Basically, we want to see if the regularly collected sample using regular approach can generate comparable results with the cryogenic collected samples. But in general, I think we would like to minimize the exposure of samples above ground to any kind of oxygenated environments, because here primarily we're targeting abiotic, reducing other minerals. So we don't want to have oxygen exposure that could potentially cause the oxidation of the minerals. So we're working on a sort of field apparatus that could potentially help us minimize the oxygen exposure during the sampling process.
Oh.
But that's still to be tested. Yeah.
Okay, great. How does organic matter in the field samples affect the analysis? So, for example, is there any potential that minerals could be coated by organic matter, and would that affect the response?
I would agree that's definitely going to affect the ORP reading, as we know that organic minerals can potentially passivating a lot of the reactive minerals. Yeah, in this case, the idea of this kit is to get an overall measurement of the system. So we're not trying to tease out different components. We're just trying to see, hey, what's the overall reduction potential of this solids, whether it's coated by minerals or coated by organics or not. So then we're trying to correlate that overall like measured reduction potential for the entire system to the reactivity of the entire system.
Okay, got it. That makes a lot of sense. So David, at this time, we'd like to bring you back online, both of our speakers, a few questions. The first is a good question from the audience. And the audience member commented that it seems really tough to distinguish abiotic from biotic degradation, given complex geochemistry and changes in the redox potential due to biological activity as well as geochemical activity. Do you have any thoughts on that? Ultimately, is it possible in the field to distinguish abiotic from biotic? So David, we'll start with you.
It is challenging because you have the confluence of particular daughter products. So in particular, ethene and ethane that can be formed both biotically and abiotically. I think the best way to sort that out is going to have to be in the laboratory where you've got controlled conditions that will enable you to create abiotic conditions or to enhance biotic to see if that is the driver. I think my feeling is that in the end, in some ways, that's an artificial distinction. What we want to know is, are the compounds going away? And can we provide a robust explanation for that? Some of which may be attributable to biotic and some of it may be attributable to abiotic. The other thing to consider with this is that we seem to want to pigeonhole biotic and abiotic, but there's a really important interface between the two, where biotic processes we know are central to the cycling of minerals in their redox states. So I think in the laboratory, we have the best opportunities to make distinctions between abiotic and biotic, but from an application standpoint, we're really looking to make a robust case for a conceptual site model that degradation is occurring and it may be a combination of biotic and abiotic.
Great explanation, thank you. Damin, did you have anything to add to that?
Yeah, I think David covered pretty well. So the one thing I want to just add in specific to the ORP measurement is a lot of these electron shuttles actually have sort of a natural origin. So the microbial can secrete some of the electron shuttle that facilitate their own metabolic processes. So that just add another layer of complication in terms of what the ORP really responding to. So, if the microbes are actually mediating some of the electron shuttle production, then potentially that electron shuttle can also help the measurement of the ORP of the minerals. So that's just another sort of layer of the complexity. But I do agree with what David just said. From the site management perspective, really important question is whether we know natural attenuation, destructive natural attenuation, including both biotic, abiotic processes, whether those processes happen in the field.
Great, thank you. Now I'll ask each of you for our audience to roll up your recommendations. What would you recommend in terms of tools to use to better understand the potential for abiotic natural attenuation at specifically chlorinated solvent sites? So again, David, we'll start with you.
Well, I think in some ways, some site managers are missing some low-hanging fruit by getting, say, complacent with pump and treat as, it's working and controlling the movement of the contaminants and maybe getting used to the idea that you're going to pump indefinitely as your strategy. What I think is being missed there is the opportunity to look for evidence that natural attenuation is occurring. In many cases, it will be abiotic, but the challenge, unlike the more simple case with biotic reductive dechlorination, is that in the monitoring samples you get, you're not seeing evidence of degradation products. So I think if you have an opportunity to look at the monitoring data and assess whether or not you can formulate a reasonable hypothesis that degradation may be occurring, then you have the opportunity to look for explanations of that. And I think the C14 assay that we've been working on is one of the tools that will give you that evidence, the evidence for product formation. I think compound-specific isotope analysis is another tool that is available. And the really nice work that Damin is doing with ORP potential is another tool. I think in the end, people have to get used to the idea that coming up with an explanation of what's going on in these cases where abiotic is a significant contributor is going to be a bit more complex than the more simple case for biotic reductive dechlorination. But having said that, the alternative might be ridiculously long pumping times.
Got it, thank you. And Damin.
Yeah, again, I try and echo what David just said. Just bring back to the biological aspects. If you look at the sort of the EPA guidance on MA evaluation framework, really there's a lot of things that have been developed for biological aspect compared to what abiotic aspects. So abiotic aspects pretty much kind of generic. You look at the iron 2 content, you look at the mineralogy using XRD, but compared to biotic, it's not that. So we know if the site has certain percentage of certain type of minerals present, we cannot say as confidently as we said for in the biological aspect, where you have 10 to the 6 deholycoides present in the groundwater, then you probably have some active natural decoration going on. So I think what David's C14 assay bring to the table, and as well as the new developed tools, can definitely improve this quantitative understanding and help to accelerate the time frame that we needed to evaluating the abiotic natural attenuation rate process. So the other aspect I want to mention here is there's sort of there's a data gap between sort of research and field. I'm really glad to see that David was able to demonstrate some of the field sample in the lab that has the comparable to rate constant compared to the field measurements. But a lot of times we're seeing the field samples has shown some degradation potential, but in the lab where we work with the pure mineral phases like pure magnetite, pure grain rust, or pure iron sulfide, that reaction rate constant is not the same as the what the field measurement shows. So I think there's also a data gap in there to understand why that's the case. So why we're not, when we work with pure mineral phase, why we're not seeing the same reaction as we see in the field data. And I think there's also a trend I observed in the recent research and development area where you have the laboratory system becomes more sort of intricately more complex, trying to simulate the field conditions. For example, there are several recent publications showing where you combine the green rust with a biochar, then you can significantly enhance the decoordination rate. Or if you have some kind of metal substitution in the mineral structure of the green rust, you can significantly enhance that decoordination rate. Those questions are all relevant in the field, because in the field, you're not going to have a pure mineral phase. You're going to have different kind of interactions with different constituents. So I think there's some area in the research arena that can further be explored to answer that question.
Great. Wonderful, Damin, thank you. And thank you to both of our speakers today for the very clear and very useful talks and for answering so many questions. Thank you both. Our next webinar in this series is on Thursday, February 24th. It will be a webinar featuring DoD-funded research to reduce the impact of waste management on base operations and provide safer and more reliable solid waste disposal practices. Registration is open, so please visit the SERDP and ESTCP webinar webpage to register for this and the other webinars through the end of 2022. Before we conclude, I would like to remind you that both the audio and a copy of the presentation will be archived on our webinar webpage in case you want to refer to them in the future. We would appreciate it if you could please take a moment to complete the quick survey that will pop up on your screen at this time. This concludes today's webcast. Thank you.